It's my pleasure to present on our drug AXPAXLI. These are our forward-looking statements, and our lawyers make me say this. OTX-TKI is an investigational product that has not been approved by the FDA or any other regulatory body. Safety and effectiveness have not been established. So what is OTX-TKI? OTX-TKI marries axitinib to our proprietary hydrogel technology. This allows the injection of a single hydrogel implant that continuously, with almost zero-order kinetics, releasing axitinib for up to 12 months with complete bioresorption. So it's kind of different than other polymers in that usually what happens, the drug is released from the polymer and the polymer remnants remain. In this case, the bioresorption occurs before the drug, so at the end, there's actually release of drug at the end, not that tail off that we often see with polymers. Why do we choose axitinib? Well, axitinib has the highest potency of all of the different TKIs that are being tested in retina right now at a picomolar range potency for all the VEGF receptors. For most of us, this is a different way to think about things. We are used to extracellular blockade using anti-VEGF agents. TKIs, all the TKIs work inside the cell to block all the three VEGF receptors as well as other receptors very potently. Slide right just shows that over time in a VEGF challenge model, so this is a model where the rabbit's constantly being challenged with VEGF, there's essentially no leakage during the time that the drug is being released in the animal. So what we have is one of the best-in-class TKIs for both VEGF and PDGF, durability up to about 12 months, a single implant that is injected through a 25-gauge needle, and importantly, this hydrogel is also used in our FDA-approved product Dextenza. So we have over 700,000 cases of Dextenza use to see the excellent safety profile of this delivery platform. So let's talk about the SOL clinical development program. There's two clinical studies, phase 3 clinical studies. The SOL-1 is a superiority study, and the SOL-R study is a non-inferiority study. The SOL-X study we just announced enrolled the first patient. The SOL-X study is an open-label extension, which we're going to follow patients for an additional 3 years after the SOL-1 and SOL-R studies are completed. And as I mentioned, the first patient was enrolled in that study. We followed very closely the FDA guidance for wet macular degeneration studies in terms of using a superiority endpoint with a 15-letter margin. We maintain masking in both our clinical studies, and we have an identical dosing schedule in the SOL-1 study. And this is very important because this is the first time that we're showing superior efficacy against an anti-VEGF agent, and the only way you can claim superior durability is to show superior efficacy. This is a SOL-1 study design. Treatment-naive patients are enrolled. They're given 2 loading doses and then randomized and then followed for 36 weeks for the primary outcome. The unmasking of the data was at 52 weeks, at which point redosing was performed, and they're redosed at 6-month intervals thereafter. What's different about this in many clinical studies that have been done in the past is this loading. So in general, you randomize at baseline. In this study, patients were randomized if after 2 loading doses of aflibercept, they gained 10 letters of visual acuity or were 20/20 vision at baseline. This is a very unique population of patients, and we'll see what happened. So the primary endpoint was met with high statistical significance, maintaining visual acuity, and this was increasing delta seen at the 52-week time point, again, highly statistically significant. But you need to look at the totality of evidence, and when you look at the OCT outcomes, first injection, second injection times, these were all met with statistical significance. This time to first rescue treatment, looking at the Kaplan-Meier curve, there's about a 24-week time point between the patients treated with OTX-TKI and those patients treated with Eylea. But this is the take-home message I want people to leave the room with. When we look at our macular degeneration studies, patients enter those studies with roughly about 57 letters of visual acuity, about 20/80. In the SOL-1 study, this is a very different patient population. They had almost normal OCTs, and almost normal visual acuity at baseline. So if we look at the quartile analysis, and you look at quartile 4, for instance, at the bottom, those are the patients with excellent visual acuity, 20/25. They gained very little vision after those 2 loading doses. With the aflibercept, they actually lost vision at that first month afterwards, but at the end, they still had 20/25 vision, so they're very happy. Look at the patients who started the study at 57 letters or so, quartile 1. Those patients are what you're more used to seeing, about a 13.3 letter gain in visual acuity at the primary endpoint. We also have our ongoing Helios studies, which are in diabetic retinopathy. We went to the FDA and got a very unique endpoint, an ordinal endpoint, not binary. So we could either prevent a 2-step loss of DRSS or engender a 2-step gain in DRSS. So either way counts for us. It is very different than all the diabetic retinopathy studies that are currently out there. So in conclusion, this is the first time we have a superiority study which was successfully met in wet macular degeneration, showing a durability of our drug of over 9 months, 36 weeks, in 75% of patients with good anatomic and visual outcomes, and the safety was good with no patients having any endophthalmitis or retinal occlusive vasculitis. Thank you.